morning students, welcome to biology classes. So today we will see that how to prepare temporary mount of onion peel for the microscopic study of cells of onion peel. So let us see what are the material required to prepare a temporary mount of onion peel. First, it is onion. All the equipments are shown on your screen. So onion, petri dish, forceps, needles, droppers, glycerin, blotting paper, blade, saffron and stain, microscope, glass slide, watch glass, cover slips. So these are the materials which is required. Now let's start. First, take an onion and cut it into 2 halves. Now we have a set of 4 watch glass. In first we have onion. In second watch glass we have fresh water. In third watch glass take diluted saffron in solution. In fourth watch glass again take fresh water. Now take a half onion and take out a fresh layer. Now you will see a thin membranous layer on the concave side. Using a pair of forceps, tear off a thin layer. Now cut a small piece of this thin layer in second watch glass in which there is water. Then after 2 minutes, transfer that thin layer in third watch glass which carries diluted saffronin solution and keep this peel into saffronin for 2 to 3 minutes so that each part of peel becomes stained. Now again transfer that stained peel into next watch glass in which there is water to remove extra stain. Now after removing extra stain, transfer that peel onto clean glass slide. And please take a precaution while transferring peel onto a glass slide. That peel will not become folded. जब आप पील को ग्लास स्लाइड पे ट्रांसफ़र कर रहे हो तो आप ध्यान रखो कि पील फोल्ड ना हो, डबल ना हो जाए. अगर वो फोल्ड हो गया तो आपको माइक्रोस्कोप में क्लियर नहीं नज़र आएगा. ओके? नाउ put a drop of glycerin on it. Right? On that section of peel, put a drop of glycerin. Got it? Now slowly cover it with the cover slip with the help of forceps. You can also tap the cover slip with the help of needle gently so that all the air bubbles will remove. Now remove extra water and extra glycerin with the help of blotting paper as shown on your screen. Now observe the slide under the microscope. You will observe a structure like this. You will observe small chamber-like structures which are called cells. These are plant cells, as these cells have vacuole and cell wall with plasma membrane, nucleus and cytoplasm. These are the onion epidermal cells. In that way, we can prepare a temporary mount of onion peel. Now children, this experiment you have to write in your lab manual with all the headings: aim, material required, procedure, observations and result. I hope this practical is clear to you. In the next video, we will discuss about further experiments. Till then, have a nice day and thank you.